The quest to resurrect dinosaurs through cloning begins with the daunting challenge of genetic discovery and sequencing. DNA, the blueprint of life, degrades over time, and its survival over millions of years until today is highly unlikely. The oldest recoverable DNA samples have been extracted from specimens that are hundreds of thousands of years old, far younger than the last dinosaurs, which vanished around 65 million years ago. Despite these obstacles, significant strides have been made in paleogenetics, a field that explores the genetic material of ancient organisms. Scientists have managed to retrieve protein sequences and partial DNA fragments from dinosaur fossils, although these are not complete genomes. Techniques such as mass spectrometry have been employed to analyze these proteins, helping to unravel the biological makeup of these ancient creatures. For example, researchers have successfully sequenced collagen from dinosaur bones, providing insights into their evolutionary relationships with modern species. The sequencing of these materials involves intricate procedures where scientists painstakingly reconstruct genetic fragments, like piecing together a jigsaw puzzle. Advanced computational methods and algorithms are used to align these sequences with those of modern animals, filling in gaps and making educated guesses about the genetic structure of dinosaurs. This process is not only technically challenging, but also requires innovative approaches to overcome the fragmentation and chemical changes that DNA undergoes over such extensive periods. While the complete resurrection of a dinosaur from existing genetic material remains beyond our current capabilities, each genetic discovery contributes to a better understanding of dinosaurs' biology and evolution. These genetic insights open up new avenues for studying how dinosaurs lived, what they looked like, and how they might have interacted with their environments. Moreover, they lay the groundwork for future breakthroughs in cloning technologies, potentially enabling the revival of other extinct species whose DNA is better preserved and more readily recoverable. The role of paleogenomics in the exploration of extinct species, particularly dinosaurs, has been transformative, providing a window into the ancient past and offering insights that were once thought impossible. Paleogenomics, which involves the study of ancient genomes, enables scientists to reconstruct the genetic makeup of long-extinct organisms, shedding light on their evolutionary relationships, physiological traits, and adaptations. This field has seen significant advancements with the advent of new sequencing technologies that allow for the recovery and analysis of ancient DNA fragments, despite their degraded state. By comparing these genetic sequences with those of modern animals, researchers can identify evolutionary continuities and divergences, thereby piecing together the genetic jigsaw puzzle that links dinosaurs to contemporary species. For instance, by analyzing the genomes of modern birds, which are considered the closest living relatives of theropod dinosaurs, scientists can infer characteristics of dinosaur genomes. This comparative approach helps fill in the blanks regarding how certain genes might have functioned in dinosaurs, including those related to metabolism, growth, and even behavior. Paleogenomics also plays a crucial role in resolving some of the most contentious debates in dinosaur biology, such as their thermoregulation mechanisms, whether they were warm-blooded like mammals and birds or cold-blooded like many reptiles. Genomic data can provide clues about metabolism rates and energy consumption that are not evident from fossils alone. Furthermore, the insights gained from ancient DNA extend beyond mere academic curiosity. They have practical implications for conservation biology and genetic engineering. Understanding the genetic basis of adaptability and resilience in ancient species provides valuable lessons for the conservation of modern endangered species. Additionally, the field of de-extinction, which contemplates bringing extinct species back to life, relies heavily on the techniques and data provided by paleogenomics. As this field continues to evolve, the possibilities it presents are expanding, promising even more profound insights into the life forms that once roamed the Earth. Paleogenomics not only enriches our understanding of natural history, but also equips us with the knowledge to potentially guide future biodiversity conservation efforts. Cloning technologies, crucial for the ambitious goal of resurrecting extinct species, have seen significant advancements since the first successful cloning of Dolly the sheep in 1996. These technologies involve replacing the nucleus of an egg cell with the nucleus from a somatic cell of the organism to be cloned, which is then stimulated to develop into an embryo and implanted into a surrogate host. For cloning dinosaurs, specifically, the challenges escalate due to the absence of intact dinosaur DNA and the vast evolutionary distance between dinosaurs and any potential surrogate species available today. Dinosaurs, having been extinct for around 65 million years, leave no direct descendants that could act as viable surrogates. Birds, the closest living relatives of dinosaurs, differ significantly in size, reproductive biology, and other physiological aspects, complicating their use as hosts for cloning. To address these challenges, researchers are exploring the potential of advanced genetic engineering technologies, such as CRISPR-Cas9, which allows for precise editing of DNA sequences. This tool could theoretically be used to modify the genomes of bird species, making them more compatible with dinosaurian genetic material, should it ever be sufficiently recovered and reconstructed. Additionally, synthetic biology could play a role in cloning efforts, particularly through the creation of artificial wombs or the development of synthetic eggshells that can support the growth and development of a cloned embryo outside of a traditional biological host. These innovations would not only aid in de-extinction efforts, but also enhance our understanding of developmental biology. Despite these technological advances, the cloning of dinosaurs remains hypothetical due to the incomplete nature of the genetic information currently available and the immense technological and ethical hurdles that would need to be overcome. Each step towards refining cloning technologies, however, contributes to broader scientific capabilities, potentially aiding in the conservation of endangered species and the study of genetic diseases, along with offering a deeper understanding of the genetic fabric that constitutes life on Earth. The ethical and ecological considerations of cloning extinct species, particularly dinosaurs, are profound and multifaceted, touching on issues ranging from environmental impact to the moral implications of genetic manipulation. The prospect of resurrecting such species raises immediate questions about the balance of nature and whether modern ecosystems, which have evolved in the absence of these ancient giants, could support their reintroduction. Firstly, ethical questions abound regarding the justification for reviving species that have been extinct for millions of years. Unlike de-extinction efforts aimed at recently extinct species where human activity was a direct cause, bringing back dinosaurs would not address a conservation need or rectify past human-induced extinctions. Instead, it could be seen as an exercise of scientific capability without clear ecological or societal benefits, potentially diverting resources from conservation efforts more crucial for biodiversity preservation. Moreover, the potential ecological impacts are significant. Introducing large predatory species such as Tyrannosaurus rex into contemporary ecosystems could have unpredictable effects on existing wildlife populations and habitats. The introduction of species even slightly removed from their original time, such as the reintroduction of wolves to Yellowstone, has led to significant ecological shifts, suggesting that the impacts of reintroducing dinosaurs would be even more dramatic and potentially disruptive. There is also the concern about the welfare of the cloned animals themselves. Cloning technologies are not perfect and often result in high rates of birth defects and early mortality. The act of cloning a dinosaur would likely require multiple attempts and lead to numerous failed embryos and potentially suffering individuals, raising substantial animal welfare concerns. Additionally, there is the risk of unforeseen consequences, such as the spread of diseases that could cross species barriers or the possible escape and establishment of populations outside controlled environments, leading to invasive species problems. These ethical and ecological concerns necessitate a careful, considered approach to any future attempts to clone extinct species, particularly those as ecologically significant as dinosaurs. The debate touches on fundamental questions about human responsibility and the limits of scientific intervention in nature. It challenges scientists, ethicists, and the public to think critically about the reasons for and potential consequences of resurrecting long-extinct species, balancing scientific curiosity with a commitment to ethical responsibility and ecological stewardship.